We tested the correlation between the amount of glucose in the growth environment of Saccharomyces cerevisiae and the expression of the PDC5 gene.
This experiment was conducted by Connor Howard
and Maddie Phillips.
Saccharomyces cerevisiae is a common type of yeast used in baking and the fermentation of alcohol. The PDC gene is a gene within the Saccharomyces cerevisiae that is involved in the fermentation process. We are using PDC5 because previous research found that it is the most consistent gene expressed among the PDC genes. PDC5 is expressed during the decarboxylation process. This occurs shortly after glycolysis during the Krebs cycle, when the yeast is grown without oxygen. It produces ethanol because of this gene. Since the decarboxylation process occurs after glycolysis, it feeds on glucose. Therefore, the expression of PDC5 is dependent on glucose.
The purpose of this study was to investigate the correlation between the amount of glucose in the growth environment of S. cerevisiae and the expression of PDC5. We hypothesized that the higher the concentration of glucose in the growth environment, the higher the expression of PDC5 would be.
In order to do this experiment, a series of detailed steps was followed. Our yeast cultures were set up using increasing concentration of glucose, a 2% concentration of ethanol, and were grown overnight under anaerobic conditions. We then extracted RNA from each culture and then converted the RNA into cDNA. We then used real-time PCR to compare the expression of PDC5 in our cultures to a normalized gene, S18. This was done so that we could determine if the expression levels were significant or not. We found that expression levels were relatively the same up until the 16% glucose sample. Both the 16% and the 20% samples showed a significantly greater amount of expression than our control sample, 0%.
Upon further analysis through t-tests on each sample, our original findings were disregarded due to the newfound statistics. We found that among the lower concentrations of glucose, there was around a 50% chance that our expression levels were completely up to chance. With the 16% and 20% samples, the standard deviation of the threshold cycles was high enough that it affected the p-value, the number that determines if our data could be said to be statistically significant.
We hypothesized that with a higher concentration of glucose in the growth environment of S. cerevisiae, there would be a higher expression of PDC5. Our hypothesis was not supported by our data, because the difference in the means of expression between all of the sample groups were not significantly different from the control group. Therefore, we accept the null hypothesis.
Our findings indicated that there is, in fact, an increase in the expression of PDC5 with an increase of glucose in the growth environment of Saccharomyces cerevisiae. Due to inconsistencies in the standard deviations of the 16% and 20% test groups, our original findings were negated and disregarded.
In order to better this experiment in the future and expand our testing, higher concentrations of glucose would be tested, possibly starting at 0% and increasing in 15% increments. Another thing that could have improved the chance that the standard deviations would be lower is if the concentrations of the samples were equalized before they were converted into RNA.
We would like to thank Life Technologies for providing us with all the necessary supplies that were needed for our experiment. We also want to thank our teacher, Henry Weeks, for all of the help with lab procedures and directions on how to use the advanced equipment.